All right. Thank you all for coming. It's my great pleasure to introduce Sam Scarpino to this quantitative systems biology seminar. Sam's the director of AI and Life Sciences in the Institute for Experiential AI at Northeastern University. He's also a professor of practice in health, both the departments of health and computer sciences, and also has appointments in network science, where he has his graduate students in that department, and then also parts of global resilience in the Roux Institutes. He is also an external professor at the Santa Fe Institute, where he's been interacting with the students and postdocs there for a number of years, and has also been integrated with the Vermont Complex Systems Center. So prior to joining Northeastern, he got his bachelor's degree from Indiana University in Bloomington. He went on to get a PhD in ecology, evolution, and behavior from UT Austin. Got his assistant professorship of mathematics and statistics and the complex systems at University of Vermont in Burlington. He went on to become the vice president of pathogen surveillance at the Rockefeller Foundation, and was the chief strategy officer at the social tech startup called the Dharma platform. And he co-founded a data science initiative called the Global.Health. And it was backed by Google and the Rockefeller Foundation. He's done some incredible work on artificial intelligence and has been applying his trade to these major questions that are addressing global health in terms of both things like the COVID pandemic and global warming. And it's published in top tier journals like Science and Nature. And we are very fortunate to have him today with us to talk about his more recent projects focusing on how to translate genotype to phenotype using artificial intelligence.
Thank you. Great. Well, thanks everybody for being here. AI has certainly been good for me because, as you can tell from the intro, I've had trouble deciding what exactly it is I'm going to work on. And so this idea of being able to use AI to link across all these various interests has been quite appealing. I created this slide for another talk, which is one of the things that I've been working on in the space of infectious diseases for the past few years, is trying to come up with ways of categorizing different kinds of epidemic curves and do that in such a way that it tells us the similarities and differences between different pathogens and how the substrate that they interact with, like human social networks, impacts them in ways that may transcend pathogen types. And so I have this slide up here that says, like, as an evolutionary biologist, I was trained to always start with a slide that had a whole bunch of different creatures on it and to say, like, this is what motivates me. And then I have a next slide, which is just a whole bunch of epi curves. And I say, like, for me, this is the new diversity slide. But part of the other thing that's been exciting with artificial intelligence is I've gotten a chance to figure out how to come back to evolutionary biology and genetics, which is one of the things that I have been passionate about for years and kind of took some time off of to work on infectious disease dynamics. But I think for most of us, one of the things that we hope artificial intelligence will be able to do is tell us something about the mechanisms and processes that underlie the differences between these diverse species, but do so in such a way that it also informs general patterns and identifies rules of life. And I think the other reason that artificial intelligence in some ways is so appealing is that a lot of the concepts that biologists talk about with respect to how phenotypes are determined from the interaction of the genome and the transcriptome and the proteome with the environment goes back to some of the earliest roots of thinking in both evolutionary and developmental biology. So here I'm thinking about Waddington's landscape of how a developmental process might unfold. And one of the reasons why I think so many people put surfaces up like this is because it illustrates so many of the challenges that we know exist when we try to make observations about living system and infer process and function, namely that we can see if, as this ball rolls down the track here, that it encounters, small variability in the friction with the track and may send it down one of these alternative pathways. And so where it ends up at the bottom may have something to do with the shape of the landscape, but it might also have something to do with stochastic effects or a whole variety. Maybe there's an environmental component and the wind is blowing to the left across the landscape, one day as kind of a metaphor. But, and I may say a little bit about this at the end, this also kind of looks like our conceptual diagram of a surface that we would be searching over in an artificial intelligence context, right? So like one of the things that we do with AI algorithms is search over complex landscapes to try to find values of parameters that work to predict one thing from another thing. And one area where I think biology has a lot to offer, although this is not what I'm going to discuss in this talk, is to actually feed back what we know about the structure, function, the mathematics of living systems onto neural networks and artificial intelligence systems themselves, right? So like a lot of AI systems are supposed to mimic these kind of landscapes, but they do so in a pretty naive way with respect to how we actually think these things look and exist in the real world. In addition, and this is one of the reasons why I really love going back and reading old papers, is there's just a wealth of good ideas. In some ways, it sort of feels like people had a lot of this stuff figured out in the 50s. They just didn't have the computers to actually kind of work, and they didn't have the data and a whole bunch of other stuff. But like conceptually, some of this was figured out. So in that same, book where Waddington introduces this developmental landscape, there's this figure of a developmental process where you have some kind of pre-tissue state that develops into some adult tissue. And what Waddington illustrates is that you have these genes that may interact with each other. And if you were to read the caption, it would say that those squares and diamonds are either environmental factors or there's some products of genes that are turning on and off or up or down other genes, and that this kind of complex set of interactions between genes and environment results in this developmental process. And so the other reason that I think AI is so useful is if it's good at anything, it tends to be good at inferring nonlinear relationships between data sets. And that's kind of what Waddington is pointing out in this figure. And it's what we absolutely understand happens as we go from genotype to phenotype, is that there's some relationship between genotype and the transcriptome, between the transcriptome and the proteome, the metabolome, all of those in the phenotype interacting with the environment. Very little of that relationship between those different layers is linear. And one of the things that we can do quite well with AI systems is handle that inherent nonlinearity. And I think for me, ultimately, and this is one of the things that I've had a lot of fun talking with many of you about today, and is what inspired an NSF grant that a lot of us have been working on, is trying to study this question in as scientifically rigorous a way as possible. And I think what I mean by that is trying to come up with a way of asking how predictable living systems are that allows us to test hypotheses, that allows us to identify general patterns, and understand something about, this figure here, that it's one thing to say genes interact with each other and they interact with the environment and there are these nonlinear things that connect all of them together, but how bespoke are these relationships for particular species, for particular phenotypes? How dependent on the particulars of the environment are these relationships, or are there general patterns that we can identify that transcend different parts of the tree of life across very different phenotypes, or not? And I think that's one of the other things that we'll be able to at least gain some purchase on with artificial intelligence. So I want to talk about 4 things today. 3 of them have to do with some science that we've been doing, and one of them is kind of where I want to take things in the future. The first is a story about how I think we learned something about the way neural networks function by trying to do comparative genomics in a very dangerous way, meaning like we took a problem that is way, way too easy to be doing with neural networks, and we did it for what we thought was a good reason and kind of learned something else interesting. And what we think that we learned is that neural networks are very good at recapitulating phylogenies. I want to talk about work that I've been doing with Brian Glazer and Vito Quaranta around linking transcription to developmental processes. And then talk about work we've been trying to do around alignment-free comparative genomics. So the other kind of theme of this is, can we find things that AI can do that we can't do with existing methods, or at least we can't do very well with existing methods? So just to get us oriented, I want to try to at least say what AI is, maybe at least to me. And probably if anyone says AI, you should always ask them what they mean by that. It's sort of like if somebody tries too hard to defend a prior and a Bayesian analysis, you can almost immediately call BS because they don't really have a principled reason beyond like it's mathematically tractable or something like that. And so if somebody says AI, you really don't know what they mean. And I think what a lot of people mean, and this is a pretty nice review article about deep learning in the biosciences. So if you want to kind of get caught up a little bit on where everybody, at least they were a couple of years ago, this is a good review. They really just mean machine learning, and that's like a totally fine replacement if you want to bump up your grant impact factor or get into a higher journal or something. But I think importantly, there are 3 categories, and some of them are more or less represented in more traditional machine learning. Like if you took a machine learning class 10 years ago or something, and those are supervised and unsupervised learning. And you're probably familiar with that. If you're not, the way that I just think about this is whether your data have labels on them or not. And then the third, and this is one that has been around for a long time, but it's kind of come back into fashion with increased compute power. And that's reinforcement learning, where you actually have the AI model learning from its mistakes and kind of improving on itself. Another way that we could think about AI, and I really like this is my favorite slide that I always put up about AI. KPMG just surveyed like a whole bunch of business leaders and asked them what they thought AI meant to their business. And so 41% of them just said it means machine learning. That's that tall bar on the far right. But we have like robotic automation, artificial virtual reality, deep learning, anomaly detection, biometrics, chatbots, digital twins, image recognition, natural language processing, constraint-based optimization. And if you're familiar with some of these, like if you're going to do a lot of natural language processing, that probably also involves neural networks. That is also a part of deep learning, although I don't actually think I see neural networks show up anywhere here. And so part of the problem with AI is that it means a whole bunch of different things to a whole bunch of different people. But I think throughout the whole rest of the talk, when I talk about AI, what I mean is some kind of neural network that we're using. From the perspective of what I think I care about most often for AI and biology, it's that I have some genome or transcriptome, proteome, some ohm data, and I have some target data that I want to predict, and I'm going to put a neural network in the middle and try to build a prediction engine for taking in as input some combination of omic data and producing as output some prediction about the phenotype or some other aspect of interest that I'm working on. And the why, I kind of hinted at this a bit, or I was pretty direct about this at the beginning, that I think one reason is that these neural networks really can help us build nonlinear mappings between the genome, the transcriptome, the proteome, the metabolome, the phenotypes of interest, and incorporate externalities like environment, et cetera. So I think in some ways they really are purpose-built for these kind of nonlinear mappings that we're all dealing with. The second is that they can allow us to scale some of our analyses to the size of many of the modern data sets that we're starting to work with. So the project I'll tell you about at the end has to do with predicting sampling location for SARS-CoV-2 genomes. And what we want to do is predict for about 500,000 genomes where they were sampled. And we want to use the whole genome to do this. Now, the SARS-CoV-2 genome is not very big. We've got things that are about 35,000 bases long. As an example, the PhD student that works on this can run the neural network analysis on her laptop in part of an afternoon. You could probably do this with, you could probably build an alignment if you had access to a supercomputer, maybe, but we would be talking days at best to get through the alignment process, and probably even longer on the phylogenetic side. Not to mention that we probably don't have the right phylogenetic models for this many different SARS-CoV-2 genomes, because I don't think we really understand what happened in the jump to Omicron and a whole bunch of other things that we just don't have good models for. Okay. First project, neural networks recapitulating phylogeny. This is work that I did with Will Landecker back well before the pandemic with the US CDC. Will is now a senior principal research scientist at Nextdoor, building AI systems for bias and misinformation, which you could imagine Nextdoor being a good place to work on that problem, if you're familiar. What the CDC wanted us to be able to do was take a influenza viral genome sequence and predict whether it would be sensitive or resistant to a class of antivirals that inhibit the activity of the neuraminidase for influenza. They wanted to do this because the laboratory assays are expensive and time consuming, and it is cheaper to just sequence the flu genome. And so could we sequence the genomes of all the flu viruses that show up and then use the neural network basically to triage the laboratory in time for evaluating antiviral resistance? This is a bit of a cartoon of the flu life cycle, but basically the way this works is that the viruses bud off, and the hemagglutinin, which is the H in the H and N's of the flu, they're responsible for sticking to the cell surface and triggering uptake back into the cell. And so as the viruses bud off, the hemagglutinin sticks back onto the cell surface, and the neuraminidase is what cleaves the virus free to go on and continue its life cycle. And so oseltamivir, which is one of the neuraminidase inhibitors, just sits down in the active site of the neuraminidase and prevents the cleavage. And so the virus is just stuck onto the outside of the cell, which is bad for the virus for a variety of reasons, because it can't go on to infect the next cell, and viruses stuck onto the outside of the cell are easy targets for the immune system to pick up on. The data that they gave us were about 2,500 flu genomes that had been sequenced and tested in the lab for sensitivity or resistance to oseltamivir. Now, in many ways, this is the worst possible thing we could do with neural networks, because the only thing we're doing here is lighting a lot of trees on fire to train the neural network, because 95% of the time, if it's resistant to oseltamivir, it's the same amino acid change over 15 years of influenza evolution. So if you just have like a logistic regression model on the amino acid sequence, you can get nearly perfect classification of resistance versus sensitive. So like we really don't need a neural network to do this. Now, originally, we have this interesting thing at Northeastern and network science that we call paper unwind, where we have faculty, postdocs, others come and like tell the real story behind the paper and how they came up with the idea, not the one that you put in print. And we haven't published this yet, but the version we put in print would not say we picked this because we were afraid that the neural network wouldn't work properly or we hadn't implemented it correctly, et cetera. So we wanted to pick something where we really knew what the right answer was, and we could be totally sure that all the neural network stuff was functioning properly. And for those of you that have played this game before, where you try to pick the easiest possible problem to make sure whatever you're doing is working, you realize you're going to spend the next 10 years trying to figure out what happened with that easiest possible problem. And that's exactly the story here. So again, we can look at the performance of the neural network. And so this is just plotting the false positive rate versus the true positive rate for the neural networks. And if you're not used to thinking about this, like being on the dash line would be, you know, just about as bad as you could get if you just sort of had an arbitrarily kind of random attempt at classification, assuming we had the data set balanced appropriately. And the faster this curve shoots up, like the better we're doing, the less trade-off we have between false positives and false negatives. And so a curve like this means that we're basically just almost perfect. And again, we should be able to be almost perfect because this is a very easy problem to solve. In fact, if I gave you 1,000 genomes, you could probably classify them faster just by doing like some kind of command line search for the amino acid in the sequence than even training any of these classifiers would take, right? Because you just even Googling, the time of Googling, finding the amino acid and doing the command line search, still going to be faster than training the model. I don't remember why we did this. We probably did it because the CDC said, well, what happens if you, instead of just like, so the way we would generate this curve, right? This is out of sample data, meaning that the model did not see these data in the training. It's the same here for the logistic regression. And the way that we would normally do this is just pick 80% of the genomes at random and use that to train the model. The CDC instead said, well, about 20% of the sequences happened after 2009, which was this big changeover in the viral lineages because H1N1 emerged in central Mexico, swept through, displacing the other flus that were circulating. So what if you just trained the thing up until 2009 and then tested it on 2009? So that's what we're going to do. We're going to train the model all the way up until the switch. Now, remember, it's exactly the same amino acid, exactly the same change that's conferring resistance. So here's model trained on 2009 H1N1, 80% of the 2009 H1N1, predicting the 20% of the 2009 H1N1 we've held out. So we're just going to take only the 2009 H1N1, hold out 80% to build the model, test it, hold out 20%, train on the 80%, test it on the 20%. And this is the kind of classification accurate, like the classification between sensitive and resistant. So all that matters is that the sensitive viruses are up higher than, or down, or the sensitive, it doesn't matter which is higher or lower, that the resistant and the sensitive boxes are separated from each other. Basically, if we can draw a line in between the boxes, that means we can classify, the model can classify between the two. The reason we're plotting it this way is that the data aren't balanced anymore. And so the classification boundary is not like at 50% or something like you would do for a logistic regression if the data were balanced. Now what we're going to do is the experiment I just showed you, which is train up until 2009 and test against the 2009 sequences. And what we find is that you can do no better than random. It's the same amino acid. So what's going on here? And we haven't published this, which is a long list of things that I should have published that we would have been way happier if we'd done this because, and part of this was we just didn't, because we weren't computer scientists. We thought maybe we were still messing something up and maybe we just didn't understand what was going on. One of the current best hypotheses for why neural networks can do what they do is that neural networks are very good at identifying hierarchical clusters inside of your data sets and learning functions to describe that hierarchical clustering and then using those functions to make predictions. So the way that I now talk about neural networks is that they just do fancy hierarchical clustering. And there's nothing that's a fancy hierarchical, there's no fancier hierarchical cluster than a phylogeny. And so what is the neural network actually doing? Well, if I train on the pre-2009 data, it's the same amino acid, but you see how there's this like big resistant branch over here, whereas in the host, like in the switch in 2009, you keep getting these resistant lineages that spill off and then go extinct, but the backbone remains sensitive. And so what's happened pre and post 2009 is the relationship between the trait and the phylogeny has changed. And we think that this happened because the phylogeny and the trait are co-evolving, right? Like if we could think about this as a speciation rate or a speciation process, that's influenced by the evolution of the resistance to the neuraminidase inhibitor. And so the neural network is memorizing the phylogeny. And if what we're doing is just like pooling all these data together and sampling 80% of them, it will see some of these kind of clusters where it spins off and goes extinct. It will see some of these kind of clusters where it predicts. And then if we throw another 20% at it, it will be just fine at doing the prediction. But if we only show it the pre-2009 data where it's much more strongly partitioned in separate parts of the phylogenetic tree, and then show it this new tree where you have all these little lineages spilling off and going extinct, it does a horrible job. Now, I think that this is potentially useful for two reasons. One, because I think it actually tells us quite a bit about how neural networks are functioning, which is kind of cool because everybody talks about how they're black boxes and they don't understand what they're doing, et cetera, et cetera. So if what we can show as biologists is what they're doing is that they're just recapitulating the phylogeny, that then actually informs what the computer scientists are saying, because, well, the phylogeny is just a hierarchical cluster. So maybe like all data sets basically have some kind of non-independence between data points. We just think in the biological sense it arises because of the phylogenetic relationship, the evolutionary process. It may arise for a whole bunch of other reasons in different systems. The second thing is it's possible, one of the things I'm deeply interested in is studying trained neural networks. It's possible that we could actually extract the phylogeny from the weights of the neural network. And if that's the case, then we can build these phylogenies a lot faster and in kind of an interesting model independent way. That's something that's a little bit further out on the edge. And then the final piece is, and this is work that we have actively ongoing now that we're calling phylogenetically aware neural networks, is can we build neural networks that aren't allowed to learn the phylogenetic pattern? And there's the way that I am approaching it now, which is just to say, maybe we can sample the data in such a way that it prevents the phylogeny from being learned. And then there's the sort of computer scientist way, which is we can just encode in the loss function some penalty term that prevents it from learning a phylogenetic structure. And then what we might like to find out is, does it then actually learn about this amino acid change? Because in principle, these neural networks could have a layer that was like genotype to amino acid. It could have a layer for the amino acid to the phenotype. Like there's no reason it can't learn that relationship. It's just, if you think about like the surface that it's looking over, memorizing the phylogeny, depending on whether you're minimizing or maximizing, memorizing the phylogeny is going to be like a very deep local minimum. And somewhere over here is the mechanism. And it might be a little bit deeper than just memorizing the phylogeny, but it might be very, very hard or near to impossible for the search algorithm to actually find that. And so instead, we're going to take advantage of the fact that we understand phylogenetics at least a little bit. We can encode that into the neural network to prevent it from finding that local minimum or getting trapped there, in the hopes that it actually finds the mechanistic relationship. Okay, linking AI to developmental processes. So as I mentioned, this is work that Brian Glazer leads, who's a biomedical informatics PhD student at Vanderbilt. And this is in collaboration with a bunch of interesting folks, including Vito Guanta, who's in the back. And what Brian and colleagues are interested in part, is studying this developmental disorder called Cornelia de Lange syndrome. And part of the reason why is that this is, a significant developmental disorder if it occurs in humans. It also involves a lot of change in transcription regulation. And so from the perspective of trying to learn gene interaction networks, it presents a number of interesting challenges. In addition, we think that it impacts the developmental trajectory of cells in a way that allows us to study the importance of historical contingency and path dependence, both from an AI perspective and also from a network perspective. So one of the things that I'm quite interested in is how we model the change in network structure over time. And if that network structure has a lot of historical contingency on it, that's a uniquely challenging problem, and one that's posed by these developmental data. So imagine what we're going to have are cells from mice at different developmental stages, wild type mice and mice carrying the mutation that causes Cornelia de Lange syndrome. And we'll have time series data. So we'll have single cell RNA sequencing data at different time points throughout the developmental cycle. So what Brian did is, can't really see this very clearly on the left, but what people do in this space is called pseudo time analysis. So what we want to be able to model is the developmental trajectory of a cell as it diversifies from some progenitor sort of stem cell like thing into whatever liver or eye or whatever it's going to become as the organism develops. While we have time series data, we don't have the ability to follow a cell as it goes through, right? Because as soon as you take the RNA out, you sacrifice the cell. So we have populations of different cells at different time points coming from the same individual. So they call it this pseudotime analysis because what you want to do is kind of try to reconstruct the path that these cells are following, but you're not doing it from observing the path of individual cells. You're doing it from observing collections of cells at different time points coming from the same individual. So if you could see this, there'd be lots of little arrows. You can kind of squint, you can see the arrows. That's the like trajectory that we think those cells are taking through developmental space. So the arrows are kind of flowing like down into the left here and then up into the middle. And so if you imagine dropping a cell there, we think as it goes through the developmental cycle, it's going to move in this pathway through gene expression space, which is going to be associated with what kind of tissue it's becoming or what kind of cell type it's becoming. However, what we want to be able to do, and importantly, anytime I say we, and you see somebody else's name at the bottom of the slide, that just means Brian, and then later we'll see Juan. So what we want to do instead is not be constrained. So right now, these are just actual individual cells. I want to know what happens if we drop a cell right here. Where is it going to go? Because what that's going to allow us to do is do in silico knockout studies where we say, okay, well, what if we knock out this gene? What does it do to the developmental trajectory of cells that get put in that part of space, which is what we think is happening with Cornelia de Lange. So Brian then trains a neural network where he takes this vector map and uses a neural network to construct a whole kind of functional landscape for that entire surface. So now I have a neural network that allows me to throw a cell down anywhere on that space. And I'm also with this neural network allowed to knock out different genes. So these are different genes. I can just knock out one of these genes and then make a prediction about how it's going to traverse through that developmental space. And so these are some example kind of simulations of like putting a cell down and watching it do its cell thing in silico as it goes through the developmental process. What we've been able to show is that the neural network can recapitulate these pseudotime analyses for both mutant and wild type. And so there are orange and red arrows. The orange is what the neural network is saying, and the red is what is observed in the actual data. And for the most part, you can't really see a lot of difference between them because the model's working. Trust me, I've seen earlier versions of this where you definitely could see the model wasn't working. And so the fact that you can't see the lines apart from each other, and in a few places you can, right? So if it really wasn't working, you'd see lots of these examples where there was like a big arrow of orange and a small red, or like a big separation between orange and red. You kind of see it happening over here, for example. So what this is saying is this is really working, which is kind of cool. And this is something unique to the work that Brian has been doing. So lots of people do this pseudotime analysis. Very few people have come up with ways of doing this kind of generalized approach with neural networks that actually appears to recreate the real data. And so one of the examples we wanted to do is reconstruct the baseline distribution of cell types and then reconstruct what the knockout version of cell types would look like, where we're knocking out a gene that affects stem cells. And so we expect it to have a major shift in the distribution of cell types. So this would be sort of like wild type or baseline, and then the colors are just clusters of different cell types that show up after some amount of developmental time in the simulations. And then if we have this big knockout, what you can see is that there's this whole mass and it's gone and kind of shifted over here to the left. And this is what the literature would sort of tell us should happen when you have this big disruption to the stem cells, is that you have a huge shift in the distribution of cell types that are left over. And so, of course, what we would like to do next is convince the embryologists to spend some money to actually engineer these mutations so that we can measure the gene expression patterns in mice with this particular knockout at these particular time points. Yeah. Counts of RNA sequences at different time points from single cells.
From like transgenic mice or humans?
From wild type and transgenic mice. I'm seeing some nods from people in Vito's lab, so I'm assuming that if you violently disagree with what I'm saying, you would have jumped in. Okay, so there's another problem though, and this is something that I've been interested in for a long time, which is the way that we generate these cell clusters is by doing a network analysis. Like if you've heard of the WGCNA analysis, that's sort of what it would look like to do this kind of analysis. But if you haven't, we imagine we can now build a correlation matrix between the transcript abundance. And once we have a correlation matrix, we can do what they call community detection and network science, which is to find groups of genes that have stronger correlations between themselves than they do with other groups of genes. And that's how we might color the different cell types here, is this kind of statistical analysis. The problem is, and this is well known, that if I just look at the learned regulatory network that shows up, it doesn't look anything like the mouse regulatory network that I would look up in the book. So I take a book and I look up mouse regulatory network, page 300, and then I take the regulatory network that I infer from the mouse cells, and they could not look more different from each other if we tried to do this. Now, there are three things we think are going on. One is that the reconstruction process is noisy. And so some of that difference is just because the reconstruction process is noisy. Two, and I think this is the most interesting one, regulatory networks are not the same in every individual. And we know very, very little about differences in regulatory networks between individuals or between populations. So probably some of this difference is that we have mice that are carrying a mutation that we know disrupts regulatory networks. And so we should not expect the regulatory network to look the same as the textbook regulatory network. However, we also know that one of the ways in which you identify genes of importance is based on their variability, right? So almost all of our statistical methods are going to be looking for some sort of variance-based test. If genes interact in a hierarchical regulatory network, which they probably do, then genes that are higher up in that regulatory network, so they're more central, they have more downstream components, there's probably less variability in expression that can be tolerated there without huge organizational effects in phenotype than in more downstream genes. And so if you believe that's sometimes true, then statistically, we're going to detect genes that are involved in the phenotypic response, but not the actual drivers of the response, because the variation is there, but it's too small to pick up with our statistical tests. So what Brian is doing is what we call the chicken foot analysis, which is one of these bad names we came up with in a workshop that has just stuck. And we call this because the chicken's foot kind of looks hierarchical. I don't know how we came up with this idea, and the idea is take the whole network, label the things that show interesting variability in expression, find all of the upstream items that are shared, see if they have SNPs, and if they do, label those as the punitive driver genes. And so Brian has done this with a neural network that input information about the regulatory structure and a variety of other things, and tries to do this analysis where we pick out things that don't show up in the statistical tests, but are on these shortest paths between a whole bunch of things that do show up. And so an example here is we have two different pathways between pairs of genes, and those pathways are labeled as important. That pair of genes is labeled as important in the analysis. And if we look, there's this IRS1 gene that is on the shortest path, meaning it's in the kind of upper part of the hierarchical network for all of these, for both pairs of genes. And so what we would actually then say is, we think that this IRS1 is actually the better candidate for maybe what's driving this whole phenotypic reorganization, as opposed to some of these downstream genes. It doesn't mean that they're not involved, but what it means is if I go in and say, do some CRISPR swapping, I might only kind of have relatively modest changes in the phenotype, unless I can go in and kind of get all of these 10 or 15 genes that are in this pathway at the same time. But if I go and get the regulator gene and swap out, maybe there's a SNP in one of the upstream regions of the gene, swap in that upstream promoter region, I actually get the whole phenotypic change, or at least a big chunk of the phenotypic change. So what do I think are the take-home messages? Neural networks are able to capture key aspects of the developmental landscape, which we think is pretty cool. With that, you can use in silico simulations to identify potential drivers of phenotypic changes, which can be used to inform decision-making around complicated and expensive laboratory experiments. And that if we can layer in some of these network-based approaches, we may be able to improve over established regulatory elements. Okay, comparative genomics, and I'll cover this part relatively quickly so there's time for questions. We are interested in taking a SARS-CoV-2 genome sequence and figuring out where it came from. We here is like the CDC or the WHO or a whole variety of other people. The way I would do this when I was in grad school would be like some phylogeographic approach where I've got a phylogeny, I've got tips on the tree of the phylogeny that are labeled with a location, and then I have a new sequence and I'll place that new sequence in the phylogeny and try to say something about where it may have come from using that approach. For SARS-CoV-2, we want to be able to do this with hundreds of thousands or millions of genomes. And we think that there's an approach that we can do with neural networks that prevents us from having to do genome alignments and allows us to do this prediction of geographic sampling location. We picked geographic sampling location because we thought it would be easy, and it mostly is. And again, as I told you, one of the now secrets that I just let all of my new PhD students in on is pick whatever you think is going to be the easiest thing to do first, and then just assume ahead of time that's going to become your dissertation, because whatever it is going to end up being super hard and interesting. And that's what happened here. So neural networks, very good at picking where things were sampled. 99% accuracy without a whole lot of training need. The question is, why aren't they perfect at it? Because we know there's a huge amount of geographic signal that's present in viral genomes. We think a big part of the reason that they're not perfect is because the data are wrong. What do I mean by that? Well, I download a bunch of genomes and one of them is going to say London. But sometimes that person in London, they got swabbed in London, but they got infected in Paris. And so the data says this is a London virus, but this is actually a Paris virus. And so our intuition is that the vast majority of these misclassifications are not because the neural network is wrong, it's because the data are wrong. And so what we wanted to try to do was kind of test this null hypothesis that it's the data that are mislabeled, not the neural network being incorrect. And so we've come up with this approach of studying what we refer to as the misclassification network. So we're interested in studying the connectivity pattern or the similarity of genomes and how they cluster for the ones that are mislabeled, as opposed to the ones that are labeled correctly, because in part, we want to be able to use this to learn something about the mechanisms that underlie the neural networks. So from a comparative genomics perspective, we could think about kind of two ways, and I've massively oversimplified this, but we have alignment-based and alignment-free methods. And the alignment-based methods would be your kind of stock standard bioinformatic approaches that we know work incredibly well, but maybe they don't scale as efficiently to hundreds of thousands of genomes, or if it's longer than 35,000 bases, maybe even just thousands of genomes. And then with the alignment-free methods, we have some alignment-free methods already, right? Like we could do a naive Bayes clustering on k-mers. So just like, little chunks of genome sequence of length 3 or 5 or 6 without any neural networks. And so if we're going to do neural networks, we have to at least show that it's better than the most naive methods. And one important thing is that oftentimes neural networks are not better than the most naive things. And so it's always worth checking that. Let's skip over this. And this, because I've sort of explained it already. So what we're going to do is take about 500,000 SARS-CoV-2 genomes that have around length 35,000. And we're going to do two kinds of classification tasks. One is binary. So we just say, okay, you have a 0, where we pick a focal country, United States, everything that's US is 1, the whole rest of the world is 0. And then we do the same thing for France, South Africa, India, China, etc. Then we'll do the multi-class classification problem, which is instead to say everybody gets their own label, each country, and we want to assign the genomes to every single individual country instead of just doing it, one versus all. And then what we'll compare are the naive Bayes method to the neural network. There are a few things that we have to do first, which is discard some places that just have very, very few numbers of genomes. So some countries have 10 genomes or something. We're going to discard those. We then need to, we don't need to, but it makes it more interesting, normalize the sample size by region. And so we'll do this a whole bunch of different times, but we'll say, okay, the minimum number is 100. And so we'll take 50 from each country, and that will be our new data set. We'll do this a whole bunch of times so that we have kind of equivalent sample sizes. You then have to pad the genomes to length and all these things. When I'm saying like we have to do something, that just means that it's what the current stuff allows us to do. And so all of these are places where we could be making improvements. We pad the genomes. So we're not really aligning them. We're just making them all be the same length. And the padding is just like putting an NA. And then we do what is in AI, they have a lot of fancy words that don't mean fancy things. And so they have this one hot encoding, which literally just means like the ACGT needs to become a number. And so like A would be 001000. That would be an A. And then C would be the next thing, et cetera, et cetera. For our k-mers, we could just have like sets of these 3 k-mers. And so we just chop the genomes up into these little pieces. Binary versus multi-class classification. Again, the reason we're going to compare the two is that we expect the multi-class classification problem to be harder than the binary classification. So we go into this saying, well, maybe the naive Bayes won't be, maybe the naive Bayes will be better or at least just about as good at the binary classification, but maybe where the neural networks are really going to shine is when we get into the multi-class problem. And then, and this is probably one of the more important slides, and so it maybe deserves more fanfare than I'm giving it. One of probably the most important parts of doing this AI work, in addition to constructing your data sets and your hypotheses, et cetera, is thinking very carefully about how you want to evaluate the model performance. And so we're going to be evaluating this across 4 different axes, which is credibility, and that's kind of the standard classification accuracy, the efficacy. And so we want to kind of do a more qualitative comparison to the ground truth, right? So like sometimes you can have really high accuracy, but maybe it's really high accuracy, but it's completely wrong about everything happening in Canada, right? And maybe we care a lot about Canada, and so it matters. Maybe it doesn't matter that it's much higher accuracy for the neural network on average, but the average doesn't mean as much for the neural network as it does for the naive Bayes. Robustness, again, we want to have results from different kinds of ways of generating the misclassification. So this would be like the binary versus the multi-class, the k-mers, et cetera. And then significance, which is what I'm going to show you is that the misclassification networks are tightly clustered geographically. We also have some results showing that the airline travel network predicts the misclassification network. We then want to have some kind of null model for generating networks that allows us to show that these networks are actually different than we would expect if I just generated random networks. And we can use something in network science called the configuration model, which allows us to have a certain number of nodes and edges connected with a certain probability and generate random networks with the same distributional properties. So here's what we find. So NB is naive Bayes and DNN is our neural network. The filtering threshold are countries that have less than that number of genome sequences get thrown out, and the sample size is the number of samples that we pick to do that normalization. And then the top one accuracy is just one way of measuring how accurate the models are. So naive Bayes is quite good, 83%. The neural network's almost perfect on the binary classification. On the multi-class, again, we see improvements over the neural networks that are significant and meaningful, but not extreme. And this is actually, again, as I said, not that unsurprising. In fact, when most people test their neural networks against very, very naive models, they often outperform only a little bit on those naive models. And some of this is because geographic information is heavily contained in the genome, throughout the genome. And by doing the k-mers and clustering them with, some sort of Bayesian clustering, it's not a very hard problem to solve. So one of the things that would be interesting would be to extend into, more complex phenotypes. So this is a little bit hard to see, but this is just an example of, like, we'll take the UK as the focal country and we'll color the probability of a sequence from the UK being misclassified or being labeled that says UK being labeled as France or Spain or Canada in the United States. And you can see that they're all kind of more tightly clustered around the UK and in Europe. And we can do this for all these different kind of focal countries and see that the geographic clustering is quite tight. And we can do this comparison to the configuration model that I mentioned to show that it actually is significant compared to what you'd expect by random wiring. And we can also show that it's related to the airline travel network. But there are some things that jump out that are equally interesting, which is that South Africa tends to cluster with lots of places. And one of the things that's true about South Africa is they have some of the best genome sampling and sequencing in the world. And so part of what's going on here is some of it's misclassification, but probably also some of it has to do with sampling strategy. So places that have a better kind of randomized sampling strategy are probably more likely to catch rare variants in their location that are also in other places and will get mislabeled at it. And so some of the mislabeling is due to real mislabeling. Some of it is actually just sampling strategy in these different places varies, and that's going to lead to real differences in your ability to do the classification. So we have a alignment-free classifier for comparative analysis among ensembles of genome sequences. We, not surprisingly, are able to show that SARS-CoV-2 has strong spatial dependency. And we think that looking at these misclassification networks can be highly diagnostic for mechanism. And so looking at things like how airline travel affects the movement of viruses, but also how sampling strategy affects our ability to identify geographic locations. So this is in the last few slides. Where do I want to go with some of this work? And my real vision, and part of why I wanted to come to Northeastern and work on this experiential AI project, and what that word experiential means at Northeastern, it means two things. One is training the next generation of decision makers to use AI in their decision processes. The second is humans and wet lab in the loop. So how do we then think about Brian's work and actually connect these in silico results back into laboratory systems, but then not stop with those laboratory tests, actually feed those results back into the AI system, right? Because we expect when you perturb an individual that part of that perturbation will be related to the phenotypic difference that you're interested in. And part of that perturbation outcome is just going to be related to that organism responding to being perturbed. And by having these feedback loops between wet lab and dry lab, you can both inform experiments, so you can do fewer experiments and learn more about the system, and also improve the AI models in kind of an iterative fashion. So we have an example here of some work that was funded by the Gordon and Betty Moore Foundation with Rebecca Carrier at Northeastern and Dan Bolnick, which is to engineer stickleback gut-brain axes onto these chips that were otherwise used for humans. So you have the whole gut-brain axis on the chip, and you can put some metabolite and measure the gene expression coming off of the chip. And so what we'll do is collect all the single cell data. We'll do the single cell transcriptomic analysis, multi-omic analysis that we talked about. We have these two different fish. If you're familiar with stickleback, maybe one of them's benthic and one of them's limnetic, and we're interested in how the differences affect their metabolic response to a different diet cue. So what we're going to do is measure the similarity or differences between these networks. This is all the stuff I've been talking about with Brian and a few of these others. Pick which of these things we think we should engineer, actually engineer them, be wrong about it, because that's the way biology works. It's probably not wrong about what was actually causative in this particular observation, but we know that, okay, let's say we know that this is what's causing, there's something here that's causing the difference. If I just swap this thing in with that thing, this is probably going to interact with a whole bunch of other stuff epistatically. Maybe it'll have some weird pleiotropic effects on other phenotypes. So a whole bunch of stuff will happen that I would never have been able to observe without actually doing the swap. And so then what we're going to do is rerun the experiment, remeasure the differences, and hopefully iteratively, but in a relatively quick way, get towards an actual causal model that's identifying different epistatic pathways that are involved, that are identifying different aspects of a historical contingency, et cetera. And that's a big part of what we're doing across the Institute for experiential AI. I'll do a quick plug. So what we have at Northeastern now, we've got about 95 AI faculty across our colleges. In the Institute, we've hired 20 research scientists and about 30 postdocs focused on life sciences, health, climate sustainability, large language models, et cetera. And we're hiring for someone that's interested in computational drug discovery or computational pharmacology. So at the assistant or associate level. So if you're interested in this kind of thing, let me know. So the takeaways, I think we have some early evidence that AI can provide nonlinear mappings between different layers of multi-omic data sets and allow us to connect that information to phenotype. We think it can scale to modern data set sizes, analyses that maybe couldn't scale previously. And we think it has a lot to do to help with single cell omics analysis. One thing I didn't talk about, although I don't know if I actually mentioned Juan. Where's the slide with Juan? Slide is gone. That's horrible. I took out part, let me just 1 second. I know I'm over time. Had an earlier part that Juan also did that involved the introduction of Juan. And then when I took this out, I removed Juan. So Juan is a PhD student who'll be finishing in my lab this year. So looking for postdocs or other opportunities, and did the SARS-CoV-2 misclassification network, but also interested in single cell omics analysis, but doing some things that are similar to what Brian is doing, but primarily what Juan is interested in is denoising single cell expression data using a combination of network science theory. So how you prune edges in a way that removes noise, but keeps real edges, and also using neural networks to just work directly off of the raw abundance count data instead of going through the whole rigmarole of inferring correlation networks and pruning and all this other stuff. Is let's just start with the actual data, model the data itself, use neural networks to help us do that, and build our analyses that way. So what do I think are the open questions? I touched on this, but I have a whole nother talk about how there's no ground truth in biology. And a lot of these AI, all these AI systems at some point assume there's a ground truth. And that's why we want to do this wet lab in a loop or field in a loop process, is that we expect to learn things about these systems that we could only have learned by observing and perturbing them. We don't really have shared data sets for model evaluation. So like we think we have this cool thing that does geographic labeling of viral genomes, but it's not like in the image or text analysis world where there are these benchmark data sets where people can test different algorithms and models to show that they really are actually doing something interesting and useful. And then I think we have quite a lot of work to do to figure out how we can link AI to wet lab and fieldwork. But I think there's just a huge amount of opportunity there. Thanks so much. I really appreciate it. Thanks for inviting me, and the great conversation so far. Looking forward to more of them the rest of the day.
Want to give trainees first